Hello, my name is David Alice. I'm a professor and head of laboratory at the Rockefeller University in New York, where my lab studies chromatin biology and epigenetics. And I'm very pleased to participate in this wonderful iBiology series. I hope that I can enlighten you today in one or both lectures of mine. What is epigenetics? Why do we care? And what is it that perhaps might be true about epigenetics such that it's not all our DNA alone? Is there more to our heredity than just DNA? I think we all grew up with the notion that real genetics is deeply founded in our DNA molecule itself. I want to use this slide to date myself and say that at Rockefeller University in 1944, it wasn't even known that DNA was the macromolecule that is the source of our heredity. That was work that was carried out by these researchers, Avery, McLeod, and McCarty. And on the left side of the slide, you'll get the sort of textbook notion that genes, the DNA molecule, really is the molecule that Watson and Crick described the structure of, that really is the basis of our heredity. And you can see in the yellow box, there are very defined DNA elements that lie upstream of genes. We often know these are transcription factors, transcription regulators. They can be positive or negative. But they tell the genes then, essentially, they instruct the genes to either be expressed or to be silent. So the idea here is, could it be genes are everything? You know, a tour de force, certainly in 2001, was the landmark sequencing of the entire human genome. It generated a lot of publicity, rightfully so, where now we had base by base information about our blueprint, our heredity. And there were many, many good reasons that the Human Genome Project was as exciting and got as much attention as it did. Genes might determine aging, and that, for me, someone as old as I am, that would be a really good thing to figure out how to improve that. Genes determine disease. Of course, that would affect many of us. And then genetic analyses might provide us a way to diagnose diseases and potentially develop therapeutic strategies. What we now know is personalized medicine or precision medicine. So these would be all good things that came from the Human Genome Project. So there are some questions that are sort of nagging that came from the Human Genome Project that I'd sort of like to address to set the stage for epigenetics. First of all, there was a surprisingly low number of genes in our sequenced human genome, only about 21,000. So that seemed remarkably small. It may seem like a big number, but it's a small number, especially when you consider that it's about the same number of genes that are in frogs and fish and worms. And I think it seems counterintuitive that we would be not including or needing more genes than those critters. Where would individual variation come from, especially if you had twins? If twins are genetically identical and they're not exactly alike in terms of their outward appearance or phenotype, where would that variation come from? Of course, we'd like to know when we transverse through our lifetime and we're influenced by outside factors, environment, diet, other things, does that influence the genome? And it would be, I think, arguably, well, the genome's fixed, so that can't really explain variation that way. And ultimately, when genes really became scrutinized with precise human diseases, was it the case that in every situation you found a genetic lesion, a genetic alteration that accounted for the disease? And if the answer was, I didn't find a mistake, I didn't find a change, how might that be explained by another property? So this brings us to the theme of what I want to talk about in my first lecture. That'll be the term epigenetics. It was coined by Conrad Waddington in 1942, and he's very famous for this contour map that depicted a ball rolling down a contour map hill. He was a developmental biologist who was interested in why in early embryogenesis can the same genome give rise to different cell types. And so as the ball rolled down either the left side of the contour map or the right side and ultimately came to lie in different troughs, he envisioned that those would be different outward appearances or different phenotypes. But importantly, again, all from the same genetic material. And epigenetics stands for the Greek prefix epi, means in addition or above or something on top of. So something on top of or in addition to genetics. So I think here's a nice way to sort of simplify your thinking. If you take a gene and think about it in terms of an English word, then it would be that's genetics, that's genes. But as we know in English, you can stress how words are pronounced. Are they pronounced quietly? Are they pronounced loudly? Do you boldface or underline words to get added meaning? Well, you don't change the letters, so the words don't change, but how you read them changes by these simple changes, and those would be your epigenes. For those of you that are more tech savvy, I'm not a tech savvy guy, you might think of genes as being the computer hardware, where epigenes are the computer software. It's the same hardware, but what you're putting into the system changes the meaning. And from this cover of Chem Biochem in 2011, a very simple cartoon notion of a puzzle where the authors are trying, or the artist is trying to show, I think, some of the exciting connections between epigenetics and very diverse parts of biology. So you can pick your favorite puzzle piece, but if I was picking a couple, to be fair, developmental biology is where epigenetics sort of was founded, and then that's the upper right-hand corner, but the lower right-hand corner, these white pieces. What about diseases? Again, nothing would be more important than disease states that might not be explained by genetics, but might be explained by epigenetics. Another very useful way to think about a genetics situation, we're almost to Valentine's Day. How about a cake? We can all bake a cake perhaps, and that'll have a certain taste. But if we literally put on different icings by our choice, it's the same cake. We haven't changed the cake a bit, but just by adding different icings, sugar, non-sugar, you might be able to dramatically change the taste. And I think that's a nice way to think about epigenetics. And it just happens that frostings normally go on top of a cake. Think of the term epi on top of genetics. More appropriately in real biology, how do you take a young embryo with early cells being established that are all, again, pretty much undetermined. They all don't know quite who they're going to be or what they're going to become. And then the same genome, as it advances through development, is going to give rise to different cell types. Blood, nerve, bone, skin. Now, once that has happened, these cells know who they are. And for the most part, they're determined. And they're going to give rise to daughter cells that carry on that identity. Blood cells will give rise to other blood cells and so on. Of course, we know now it's a very hot area of biology. There are small populations of cells we refer to as stem cells that are held in reserve, and they seem to have very much interesting potential because they are pluripotent. They can now still go into different cell fates. And a very hot area, of course, is can you take a fully differentiated cell and reprogram it and get it to go backwards into a more pluripotent state. But the big point I want to make is that all of these cell types, no matter what cell type they all have the same genetic material. And sadly, I think very closely connected to stem cells, when you reprogram and you sort of make mistakes in this epigenome layer, you might actually be able to sort of push those cells backwards in development where they would take on a more undifferentiated property and take on more proliferative capacity. And there seems to be many, many examples now where those type of cells take on a cancerous phenotype that, of course, is often a very dreaded disease. So how do we really start to think about epigenetics now? Not so much with the cell biology, but how about the actual mechanisms? Now, this might make you think he's crazy. This is, I think, a wonderful cartoon that was drawn by one of my former lab members, Sean Taverna, who's now a faculty member at Johns Hopkins Medical School. And with apologies to the women who might be listening to this, the DNA molecule is clearly stepping out of the shower, and that DNA molecule is making a choice. Should it grab a towel that's labeled histone, or should it grab a towel that's labeled herstone? And it looks like nature opted for the histone, so with apologies. So what do I really mean when I say histones? You go to a schematic drawing of the nucleus. Our DNA in higher cells is not naked DNA. It's in fact packaged by these proteins that kind of give an impression of a bead on a string. These fundamental repeating units are termed nucleosomes. And what nucleosomes really are histone proteins that are acting as a scaffold around which roughly 2 turns of DNA wrap. And then you can see on the schematic cartoon, these beads on a string or nucleosomes will wrap further and further into higher order structures that truthfully are still being determined. But if you take a step back and you think about this structure maybe like a slinky toy, you might be able to understand right away that if you were to pull that slinky apart or find more what we term open or decondensed chromatin, that might be a better environment for genes to be expressed. Where in contrast, if you took that slinky toy and just compressed it, more like sort of all of that chromatin that's really compact together, that would be a harder environment for genes to be expressed. Even these transcription factors would have a harder time finding their DNA sequence to act upon in that really compacted environment. And under some conditions of biology, and it's important for me to show you a real nucleus. This is a human cell nucleus stained in such a way that you can see dense and decondensed chromatin. This is an electron micrograph. And I hope you can see there are very patchy, very dense domains that are within that nucleus that are termed heterochromatin. That would be this compacted chromatin environment, where interspersed around these regions are more open, a decondensed chromatin that's referred to as euchromatin. So here's a real micrograph of a real human cell. And now under some conditions you can actually tease out that chromatin, float it if you will, outside of the nucleus. And you can see now these chromatin fibers sort of in their nearly more visible states. On the lower panel you can see this sort of classic bead on a string, like a pearl necklace if you will. And somehow that chromatin, that more euchromatin, open chromatin, may be able to be flipped into a molecular switch, by a molecular switch that'll compact that and turn that into the top panel, which is more condensed, compacted chromatin. So of course it became important for my type of lab, a biochemistry lab, to try to ask, well, what would those molecular switches be? What would be the target of this sort of switch that could throw on and off states between chromatin, euchromatin, and heterochromatin? So this takes us to another Rockefeller, I think, very pioneering discovery. Here's a nice look at this individual I want to pay credit to, Vince Alfrey. And you can see that the nucleosome unit now in atomic structure, you can see in different colors the histone proteins wrapping DNA. I think you can just see some of the Watson and Crick helix wrapping on the outside of the DNA. But you do see these tail projections that are emanating outside of the DNA helix. And what Vince Alfrey, who's shown in the cartoon, who's shown a picture of, proposed in 1964 was that there would be chemical modifications, chemical marks that would be added to these tail domains that would then act as a switch. They would be a signal, if you will, that could instruct the cell to either be on or off. So it was his dream, his hypothesis, that these chemical modifications would potentially be that switch that I was referring to in the past slide. Now, of course, every chemical modification would have potentially the need for an enzyme system that would add that mark, and in my lab we've nicknamed these writers, or there could be an opposing enzyme system that would strip these chemical groups off, which we've nicknamed erasers. And they would oppose, they would be the on-off, if you will, switches. And this was Alfrey's hypothesis, literally proposed over 50 years ago, 1964. Now, how would you access these enzymes? I think many of you would not have thought about this model organism. So on the top panel is a schematic cartoon of a ciliated protozoan that runs its life, remarkably, some very cool biology here, with 2 nuclei. And a real picture of the tetrahymena is shown below on a stain, so you can see sort of the organism on a slide. This is a unique property of ciliates, that they run their life with 2 nuclei. The larger, and hence the name macronucleus, is the nucleus that's solely responsible for gene expression. This genome is packaged in a way that you might think of it as a massive euchromatin. And in contrast, the smaller, in this slide, micronucleus that's shown in red, it is very compacted. You might be able to tell that on the slide on the bottom, and it is completely transcriptionally silent. What's important about that is that nucleus does no transcription. It's reserved for an important feature in the organism that isn't really displayed here until it has the sexual life cycle. So you might think it's not even necessary, and that would be true here. It doesn't play a role in this case. Now, when I was a postdoc, I was intrigued by this system. I thought it was a little bit strange to work on something so odd, but I want to show you one gel of mine. So this is prehistoric, and what it is to the best of my ability, I turned to trying to biochemically separate macro and micronuclei. And I joined a lab, Martin Gorofsky's lab, that was able to do that. They had pioneered those methods. And what you can see here are two lanes of a gel. On the left, you can see the macronuclear histone proteins displayed, and on the right, the micronuclear histones. And I hope you can tell, without going into the details of the gel system, there's a much more laddered sort of property or look to the macronuclear histones, and I've sort of tagged some of those ladders, those rungs of the ladder, with these green dots. What was striking is you saw a lot more laddered effect, if you will, in the macronuclear histone preps that I made, and you didn't see nearly as much of that ladder in the micronuclear preps. That sort of box, that dashed box, shows you where you don't see many of the laddered forms. So I thought this was pretty exciting, and I thought this might give me a handle on using this organism. What I need to now explain is that those ladders are due to charge differences, and each rung of the ladder is due to an increased chemical group, which was exactly what Alfrey proposed, which were these chemical groups called acetyl groups. That's too much detail maybe, but that's what I thought then would give me a good chance to go into the laboratory, purify the macronuclei, and isolate the writer that would be responsible for adding acetyl groups to the histones, as you can see here, primarily in macronuclei. So I started my lab group. I was able to attract a few brave students into my lab, one of whom was Jim Burnell. And this was actually right from Jim Burnell's PhD thesis. Jim's long been out of my lab, but it was Jim's dream that he could sort of invent or use, I think, a quite ingenious in-gel activity assay that I'll quickly describe to you. Jim's idea was to put histones, the real proteins that I've been describing in my lecture, into a gel. So that's highlighted by the blue color. So he impregnated an entire gel with blue histones. They're not really blue, histones diagrammed in blue. And as a control, he put in another identical gel, non-histone proteins that would be a control. And then the key was to take extracts from this macronuclei, from Tetrahymena, and run those out onto the gel. And in a perfect world, it was his dream that if he then sort of treated this gel with steps that I won't necessarily explain well, and then importantly, add the radioactive cofactor of the reaction, which some of us would know as radioactive acetyl coenzyme A. So the acetyl groups come from a metabolism, and he would do this, incubate this gel, wash it, and then in a perfect world, he might actually see where the enzyme is as this red band with the arrow. Get over there and point to it. And in a perfect world, you wouldn't see that with the other control gel. And this is actually the very first gel that Jim produced after giving this a try. I hope you can see a very strong band on the left that was in the histone-containing gel. So pretend that that was the gel impregnated with the blue. And to the right is an identically treated, identical sample that was, I think he used a protein called BSA, bovine serum albumin. It's not a histone. The idea was a histone writer wouldn't work on that, and there was no strong band there. It's to Jim Burnell's credit that he then purified enough of this P55 band from 200 liters of tetrahymena to get enough of this protein sequenced and the gene cloned, and it came to perhaps be one of the things that my lab is best known for. Jim's mystery band turned out to be, shown in this cartoon, look at the top half, a histone acetyltransferase, or HAT, that was already known in yeast experiments to be a positive transcription regulator. So what was not known about this positive transcription regulator was that it was an enzyme, that it was a histone acetyltransferase, or HAT. So that was really starting to now galvanize certainly our thoughts and the field's thought that you had to pay attention to this chromatin problem. Just a year, Jim's paper was published in March 1996, so remarkably 20 years ago, and another lab the month later at Harvard University, completely independent of us, Stu Schreiber's group, and a graduate student there too, Jack Taunton, found through a really ingenious chemical strategy an enzyme activity that would strip these groups off. It would be the opposing enzyme, and it was referred to as a histone deacetylase, or HDAC. And remarkably, again, this protein was already known in yeast to be a transcription co-repressor. So already the magic had happened. Our protein was thought to be a positive transcription regulator. The Schreiber-Taunton enzyme was thought to be a repressive nature protein. And what they both were doing was writing or erasing acetyl groups from chromatin. Because I think this is the thing that my lab might be best known for, I wanted to introduce you to the real Jim Burnell. How I think of Jim Burnell is on the left, the before. This is how he looked every day in my lab. He always went into the cold room with a hat because he was going after a hat and it was cold. But he actually won in 1997 the International Prize for the best PhD thesis in the world. And he was flown to Stockholm, Sweden to rub shoulders with the Nobel laureates. And my question for you is, where do you think Jim looks more comfortable? It's kind of like me standing here. I think he's much more comfortable in the lab as a lab rat. And I always have sort of made fun of Jim for that, sort of almost, I don't look very happy getting his award. It's a footnote that's unimportant to you, but I just think science has to be fun. Jim's paper was published in this journal, Cell. It was published on March 22nd, 1996, and that happens to be my birthday to the day. So that was pretty cool. So let's have a little quick summary. If you followed me to this point, classic genetics, I think, is illustrated on the left. We have bona fide important DNA molecules that really are the kingpin of our heredity. And that material is relatively fixed. It can't be altered. And of course, they're gene regulators that recognize DNA sequences, and they really do quite a bit of the activation. No question about it. What I've been trying to talk to you about is a much more flexible fluid system, the epigenetic side of the coin on the right. And the point would be that these chemical groups, no matter what they are, they can be added, they can be subtracted, and they give the genome a very important way to be responsive to what's going on rapidly. But most importantly, if you look at the DNA molecule, should a mutation occur, tragically, whatever, something that you truly inherit from mom or dad, but a genetic mutation, we don't have good ways to fix that problem. And should that be a disease-causing mutation, we're sort of stuck with that. In contrast, if any of the epigenetic modifications, the landscapes, we like to refer to this as an epigenetic landscape, should those be set up and misestablished, then there are ways to think about reversing those problems because the DNA has never really been mutated. The DNA is fixed. Remember, this is a problem of what's above the DNA template. So as soon as these enzymes began to be recognized, these writers and these erasers, many labs began to try to target them for drug development. One was very successful right across the street from where I am now at Rockefeller. That's Memorial Sloan Kettering. And in a group led by Paul Marks, he was able to sort of begin to pioneer putting these epigenetic drugs into people in clinical trials. This is a patient that was treated in Memorial where pre-treatment is shown on the left. This is 2 different scans of this individual's chest cavity. I hope you can see these arrows point to large tumor masses that were in this individual's chest. It actually was a cancer of the larynx that then spread to the lungs. And after 8 weeks of treatment on the right, it's the same individual, where I hope you can see that these tumor masses are either almost gone or much more hollowed out. It's certainly a much better situation for the patient on the right. And these kinds of very promising things actually led the Marks Laboratory to then develop this to such a state that this actually has become one of the first FDA-approved drugs to treat a specific kind of cancer. Now, this one may be a little hard for you to see because it's sort of an internal scan of the body, but also at Memorial, here's a woman that was treated for a skin cancer. This is before the epigenetic drug therapy. And just quickly, I think it's quite easy to see in visual, this is that individual after treatment with these, we'll call them epi inhibitors. So again, I think, and now this is, of course, spawned worldwide interest in these enzymes as being very powerful drug targets, and they're getting scrutiny worldwide, in part for reasons like this. So to sum up this portion of my eye biology talk at this point, now whatever your favorite modification is, think of it as there are classes of enzymes called writers, there's classes of enzymes called erasers, and now we even know that some of these chemical modifications can literally become read by reading modules that read the modification and plant themselves down on the histone tail in ways that bring about either chromatin opening or chromatin closing. And to be accurate, to this minute in time, even these readers have become successfully drugged. So every part of the language, if you will, reading, writing, and erasing have become now attractive drug targets, some of which are being clinically used. So what I have neglected to tell you about, I think, is more on the silencing system. So another chemical modification that Alfrey proposed in his original 1964 studies is maybe there's different modifications that could, again, act different ways. I've talked to you mainly about acetyl groups as a very promising on mark, but what about methyl groups? That's not a very different chemical group, but how was the field going to get interested in methyl groups? Well, this is an elegant epigenetic biology shown here. It's a classic case where, once again, the take-home would be you don't change the DNA, but you alter the phenotype. So this is a Drosophila eye. I think it's very easy for you to see on the right side, 2 different fly eyes. One is very red, one is very white. And what's happened here? Now, I think this is odd, but I can't make this go away. There is a gene in Drosophila called the white gene. I'm pointing to it. It's on the tip of the X chromosome, and I think it's a little odd and confusing, but fly people name their genes based on their mutant property. So when the white gene is active and wild type, I hate to say it, but it makes red eyes. So that's just kind of annoying. But when that fly chromosome has been treated with X-rays, and that white gene has been flipped, and that white gene, without any DNA sequence changes, lands next to heterochromatin, that gene silences. So that concept is called position effect variegation. And it's a good name. It's a positioning of a gene into a new neighborhood. You didn't change the sequence, but clearly on the right you can see those fly eyes go from very red to very white, and sometimes it's not that black and white and it variegates, and that's hence the name position effect variegation. Now, because this was Drosophila, researchers realized they could look for mutants that didn't do this position effect variegation very well. And I want to only tell you about one that was hugely important in the field of epigenetics. Let me ask you to look at the bottom of this box where you see something called SUVAR39. That means it's a mutation that suppresses this phenomenon I've been describing, position effect variegation. And what's the SUVAR39 part all about? That stands for, it was a mutation that was mapped on chromosome 3, no big deal, and it was mutant number 9. Okay, chromosome 3, mutant number 9, and of course the important part is what did it do? What was its role? What was its molecular properties? And I get to tell you one of my most favorite sort of stories in my career was when the individual shown here in the lower right, Thomas Genuine, literally obtained some evidence that the mouse homologue of Suvar39, not the Drosophila, but the mouse, smelled like it was a methyltransferase writer. And he wanted to team up with a lab like mine and see if we could figure out if that was so. And so using a lot of the same sort of approaches that Jim Burnell and we had done with acetyl writers, we teamed up with Thomas's lab, and shown here is the blackboard from my office to this day down the street. And the biochemical take-home message was that the mammalian Suvar39 was indeed a methyltransferase. That means it's a methyl writer. And remarkably, its substrate, its target, is H3, histone 3, at what position? Lysine 9. Unbelievable. So this Suvar39 was named correctly all along. And I think Thomas and I still, we're very good friends. We laugh about this. You know, it was just unbelievable that the geneticist named this right all along without even knowing what it did. So what's important? I'll give you sort of now a raging insight into what people now think. On the left side, it looks like there are situations where you want to activate genes. You might do that by taking these acetyl groups, enzymatically adding them with a histone acetyltransferase writer. Now we know that once that mark has been written, there are proteins that read that mark, like shown here, protein X. They'll dock on that acetyl group. They'll bring about gene activation. And when you really want to shut that gene off, all you have to do is employ those HDACs that'll strip that acetyl group off. And so that's a very sort of attractive, well-worked out paradigm in the field. In contrast, let's take a situation where you want to silence a gene epigenetically. No mutations, just silence the gene epigenetically. Well, in this case, you're going to have to make sure that that lysine residue, that's the blue K in these slides, that's the one-letter code for lysine. You're going to have to make sure that you don't have an acetyl group there. So you might employ the HDAC to remove the acetyl group as step one. Then you'll go to the histone methyltransferase shown in red, and you'll add a methyl group. So that's this HMT, that's the methyltransferase writer. You might think of that as the Suvar39. And then once that methyl mark has been put on, there'll be readers, like shown here in Y, in red, that literally silence and compact that chromatin by reading the methyl mark. And I have to say, it's funny to me that when this slide was made, I thought it was pretty nice, but we didn't know, and I don't show on this cartoon, was there a demethylase? Was there an enzyme that took the methyl groups off? And now indeed Yang Shi and his colleagues and now many others have found that even these methyl marks can be enzymatically removed. So in this sense, this slide is even out of date. But nonetheless, acetyl marks generally track onness, and methyl groups generally track offness in chromatin. So a couple biology examples. Here's another nice cartoon. It wasn't drawn by someone in my lab. You can see that it's a mom and a dad doing their thing, but the sister's clearly irritating the brother by saying her epigenome. Now I put in the epi. Her epigenome is more complex than his. And I think that this is a case where she's right, because every woman has two X chromosomes. So that's underneath her, the two pink chromosomes, two X chromosomes, where every male, young boy, father, whatever, XY. So, how does nature create a balance on those X chromosomes? There's two in the female, there's one in the male, and what they've learned to do is to literally shut off early in development one of those X chromosomes. That's called X inactivation. And you'll think I'm making this up, but when you take female mammalian cells and stain them, there's an example of two. You can't see much staining on the left panel, but if you stain with a methyl antibody, that's a methyl histone antibody, I hope you can see there's little patches that light up red. And those are the completely shut down heterochromatic inactive X chromosome. If you did the same staining experiment in a male cell line, you won't see any of that. And as a footnote, if ever you've seen a calico cat shown on the right, that coat color patches is all a reflection of female exon activation in the cat that happens to be tracking to a coat color gene, and that happens early in development, and it makes different coat colors and different patches, hence the calico coat properties, and calico cats are always female. They're female because of this coat color phenomenon that tracks back to exon activation and methylation. Identical twins, I mentioned, we should know that they are absolutely identical genetically, but I think if any of you know identical twins, they're often not identical, and it's becoming an attractive model system, not these two individuals. When people have studied different behavioral properties, different disease properties from monozygotic twins and found out that they're not identical with respect to their epigenome. And that now becomes a nice model system for identical genetics, but not identical epigenetics. And then, of course, I certainly grew up thinking that if I wanted to be abusive to myself, that's not so cool, but at least I'm only hurting myself. So it was a life choice that I was making. And I think a lot of people might have said, we are what we eat. But much more recently, because of this revolution in epigenetics, others have written other alterations of that quote that might be, we are what our parents ate and maybe our parents before them. And I'll give you a nice visual of that. These are identically genetic mice. But the only thing that was varied in this experiment by these researchers was the diet that their mother was given when these babies were in utero. So the question was, wow, I just altered the diet of the mother. In too much detail, these were alterations in a diet that would foster the methyl modification. And you can see that the different pups, all genetically identical, are phenotypically very different sizes, very different coat colors. And that's kind of a scary thought. They're identically genetic in terms of genetic terms, but the diet of the mother impacted the outcome of the children. And if you take this to one more extreme level, Michael Meany and his colleagues have been asking questions that fall more in the realm of social behavior. And that would be, what about having a model system that might mimic good moms versus bad moms? And he's got mice that actually are very, very good moms, meaning that they lick their pups a lot, they kind of hold and cuddle their babies quite a bit. And he's found some strains of mice that are just the opposite. So we'll sort of call them bad moms. They don't cuddle, they don't lick. And he's then asked, what happens if I can expose genetically identical mice pups to good moms or bad moms? And as you might guess, he's now paying a lot of attention to epigenetic marks, and he's finding out that the marks change in the brain regions that he studies that are responsive to ultimately alterations in gene expression that lead to alterations in stress and inherited phenotypes. And it's quite amazing to then think that this may be socially relevant in our society when we have unfortunate situations that track good moms versus not so good moms, not only in terms of the tragedy of it all, but also the upbringing of children. And perhaps can traits be passed on to even their children that are inherited through this epigenome layer. So this would be a summary slide. I think you've seen it before. I want to leave you with the impression that on the left side is the classic textbook genetics. There's nothing wrong with that. And in fact, it's very clear that some things that are like smoking and too much sunlight, they can indeed alter the DNA, mutations that can cause disease. And that's certainly something we do want to protect and hopefully inherit good copies from mom and dad. But there is now an emerging, if you will, revolution in an understanding that genetics doesn't explain everything. And there seems to be, in hindsight, you might think, a much more dynamic opportunity for these genomes to be responsive much more quickly in time to needs that might be epigenetic in nature. I should mention for completeness, it's not just the histones that are chemically modified, the DNA as well. And these things are now a raging topic for biochemists like myself. again, writers, erasers, readers, and what's really rapidly ramped up the interest in these modifications is that many, if not all of them, are now associated with human diseases, notably cancer, and many are being targeted for drug development. Why? Again, because the genome is relatively unfixable. The epigenome is much more responsive to being fixed. So if you're interested in this topic, I'm not trying to plug this book, but a pleasurable sort of challenge for, it's a complicated field. So some time ago, Cold Spring Harbor approached a small number of us to write a textbook on the subject. And if you're interested, this is the second edition of a textbook that was just published, came out this last year. And importantly, it makes me sort of want to introduce you to the editorial team. You might recognize Thomas Genuine on the far left because he's actually the scientist that we enjoyed working with on the methylation and the Suvar 3.9. That's been a long-lived friendship. And Monica Lochner, Danny Reinberg, myself, and Mary Laura Corporas are all part of the editorial team, which helps me make the point that besides your lab mates in the lab, it's equally a privilege and fun to sort of carry out worldwide interactions and collaborations, and in this case, an academic project with so many talented people. So I'm done with my first lecture. I would be remiss if I didn't thank many people. Of course, anything I referred to, I showed you a few lab people's pictures like Jim Burnell, but I've had a rich career of many, many talented students and postdocs and technicians. We don't do many of our projects without many important collaborators. The iBiology staff here has been wonderful. I'm not used to being filmed or any of these productions, but they've treated me really nicely. The Lasker Foundation for allowing this to happen here, and my funding sources, Rockefeller University, and you all for your time and attention. Thank you very much.